All right. Thank you so much. And thanks for the invitation. Let's see. Laser. Ready to go. Okay. So we developed a inflammatory clock of aging that was published in 2021 in Nature Aging. So maybe if you have samples before and after anotics or liminin, I'll be happy to put you in contact with the company that's commercializing that one. So today I'm going to talk about something different. Organoids in 0G, and this started during the pandemic when Susie Sanello, who used to be at the Human Research Program in NASA, reached out to me and she said, David, your research in the immunome and in aging is so appealing to us because we're trying to resolve all these issues that have astronauts. So try to develop countermeasures for accelerated aging we see in our astronauts, or protect us. And I was shocked. I didn't know that astronauts suffer from accelerated aging. I wanted to be an astronaut when I was a kid, but I had no idea that space and microgravity accelerate aging in different ways. And I became good friends with Susie. We got NASA grant together that went to the Buck Institute later. She passed away. I realized that she was suffering from a pancreatic cancer. She passed away a few years back. And I took that torch and continued the research that she started with University of Washington now and NASA. So much of what we would discuss were how astronauts suffer from this accelerated aging form. It's basically a natural experiment in healthy people that go in space and have neurocognitive issues. They have five-fold increase in developing heart attack during their life course. They have immune reactivation and many other derangements that happen with aging, but this happens at an accelerated pace, 30-fold increase-ish. So what we were discussing with Susie are the three major risks that NASA identified: sensory motor derangements, cardiovascular deconditioning, and immune reactivation. And so my research basically shifted. Half of my lab now is working on space medicine. And we published 5 papers all together in the Nature package and some of the Nature journals and Nature. We got the cover of Nature in 2024 on the space omics, comparing space flight and microgravity in the lab and seeing how these things can drive accelerated aging in humans, in real humans that go in space, but also in the lab. Can we envision a way to simulate microgravity? And we did so. So for the past 3.5 years, I've been working on organoids in the lab that basically get subjected to simulated microgravity using a NASA-designed instrument that's called the rotating wall bioreactor. That's right here. So we're using 3 types of organoids. 2 of them are iPSC-derived, cardiac organoids, brain organoids, and immune organoids from tonsils. So tonsillectomies, mostly from people with sleep apnea. So we subject these 3 types of organoids to simulated microgravity for 1 day. And when we do so, then we do bulk RNA sequencing, and we do functional experiments, and we map that to aging signatures and disease. And that way we can possibly identify different geroprotectors and also mechanisms of aging during this window of time. So our reference cohort is the GTEx. GTEx is a large repository of postmortem tissues, 54 different tissues in about 1,000 people. Many of them have also paired blood. And the age range is 21 to 75. So we have a decent age range. With that, for each one of the tissues, we can have whole transcriptomes across the age range. And so we can build clocks for the different organs and compare that to our organoids. And so when we do that, we can validate the neuroorganoids because they resemble the brain, the human brain, the most in their transcriptome. And the cardiac organoids resemble the heart, which is a really good thing that we have that validation. In the GTEx repository, there's no tonsil data, so we couldn't map to tonsils, but the closest was blood and spleen. which is telling us, tonsils are good proxies for the human immune tissue. So then we went ahead and compare microgravity, so our system, against flown organoids that were flown in space. These are different studies that were done with, say, brain organoids, heart organoids, and PBMCs in space. And the way to read these plots is basically this is a rank plot. So if you see warm colors in these two quadrants, that means that this study here and this study here are both going in the same direction. The genes are changing in the same direction. So both up and down genes, they are concordant, and the p-value is right here. So if you see warm colors in these two quadrants, that means that there's an opposing signature. And I'm going to get back to that when I look at the tonsil organoids that we stimulate with vaccines. We can actually take these organoids. The brain organoids are, they're firing. They have activity. The heart organoids, we're testing heartbeats, and the immune organoids, we're stimulating them with the flu vaccine, and they're generating an antibody response. So as far as hallmarks of aging goes, the neural organoids are mostly affected in their cell senescence, deregulated nutrient sensing, loss of proteostasis, and that maps to Parkinson's disease, I'm going to show you in a moment. Cardiac organoids is mostly loss of proteostasis and mitochondrial dysfunction, and cell senescence as well. That seems to be universal. And then you have the tonsil organoids with cell senescence and chronic inflammation for the most part. And then some other hallmarks of aging that get accelerated with microgravity. Then we took the biological aging clocks. We have clocks for neural organoids, and as you can see, these are 1G, this is just the control, and you can see about 10 years biological aging increase in 24 hours for brain organoids. For heart organoids, it's about half, so they're more resistant, and I think it comes down to having immune cells that are buffering the effect of microgravity. I'm going to get to that in a moment. So for tonsil organoids, we don't see that age acceleration, but we do see a negative effect in the function of those tonsil organoids. So mapping things to diseases, you can see here Parkinson's disease is one of the top enrichments. You see many genes that participate in Parkinson's disease that are elevated or changing in microgravity. We validated that in RT-PCR. We also did functional assays in cardiomyocytes and these organoids. Cardiomyopathy seems to be the disease that we're mimicking the best. And what you see here is a huge change in the contractility of these hearts, or mini hearts. They're so cute. It's like 1 millimeter, and they're beating. So you can see in microgravity, when you have endothelial cells in there, you have a massive decrease in this effect. When you don't have endothelial cells, that doesn't happen. So we know now that certain genes in endothelial cells are causing the effect of, that we see in microgravity. Then when we take the tonsil organoids and we put them in culture in the presence of the live attenuated flu vaccine, they produce antibodies. And they also are able to mount an immune response in vitro. What you see here is a really strong negative effect when you compare the microgravity and 1G and the live attenuated influenza vaccine versus unstimulated, which means that all of the genes that are changing, either increasing or decreasing with the live attenuated flu vaccine as a response to that vaccine, they are, the effect is the opposite with microgravity. We have a model for immune aging that we can now use to target these effects and identify geroprotective agents. So we're screening a lot of different compounds now to see if we can reverse, and we tested one that was published in the Nature Communications paper, quercetin, just as a canonical one, and worked pretty well in reversing 70% of the differential expressed genes in immune cells. And this is just to show many of the genes that are upregulated with the flu vaccine are downregulated, basically miming this part here, right? with the microgravity. This is a busy plot, but what I'm trying to show here is that the effect of microgravity is much larger than the effect of aging. So this same gene, MT1G, is right here. This is the effect of microgravity. This is the effect of aging in the brain. This is real brains that were from the GTEx data. This is the organoids data. And so that replicates along the way. And what you can see is that the effects in microgravity are much, much larger. So we have a really strong model, or platform rather, to study these diseases of aging and be able to screen for different compounds, see if we can either reverse or prevent these effects. One thing I should mention is that when we put the cells back in 1G and we do time series analysis, you can identify naturally occurring compounds that the cells produce to rejuvenate. So there's say, an improvement, an adaptation when you put the cells back in 1G. And so we're at the moment analyzing this in a time series fashion, and we're looking at really interesting compounds that are produced by the cells, not external, but endogenous compounds that we can now see if we can replicate that in our model. So I'm going to talk about a way to make money out of this, because it's really hard to make drugs. And trying to raise capital for this company for about 3 years now, 2.5 years, and it's hard. One, because it's a very it's a high-tech, new technology that people are not used to see. The FDA Modernization Act 2.0 in 2022 is pushing for the use of organoids, but the whole industry is behind. And VCs want to see preclinical data in animals, which is terrible. But it's hard to raise money on that. So I said, why don't we just use blood and see if the blood accelerates aging in humans, and we can maybe leverage this model for longevity medicine and apply this as a wellness test. And so we're basically trying to answer the question and tackling the idea that trajectories matter. So what I'm showing you here is iPace, is one of the clocks that we built a couple of years back. And you see this individual here, you see, it's a calendar age and iPace age. It's pretty, I would say, protected, or it's in good shape. You see this one here has accelerated aging, have around 10 years accelerated aging. So you would think that person is at risk for developing these diseases. Actually, when you have the longitudinal data, and that's the beauty of doing longitudinal studies of age, like the 1,000 Immunomes project, you can see that this person accelerated pretty fast and died out of heart attack. This person here was even decelerating slowly and is still alive and is doing well. So trajectories matter. Having a one snapshot doesn't give you the whole picture. You need to know that trajectories, and how can we take cells today and project that into the future? Can we see that future happening if you have cells in microgravity? And so we did an experiment, and we had cells from individuals that were longitudinally profiled before and after 5 years. We took the baseline cells, we put them in microgravity, and they mimic the 5-year later data point. What you see here is almost 95% concordance between baseline microgravity and older cells. So this is real aging that happens. This is not a biological aging clock. This is real aging that happens in these individuals. We put the cells in microgravity, and we can bring the cells into the future. It's pretty remarkable that we can now map these things and understand your future aging and say which hallmarks of aging will affect each individual, what are the different interventions that could work for that person, and how do we do that? Same story, by accelerating aging in microgravity, 24 hours, lysing the cells, sending for sequencing. We work with Novogene, And now we're reporting with an incredibly powerful platforms called the NU. One of the CEOs here he'll showcase part of this work. And just to recap, this is the random position machine we're not using. It stresses cells to a large extent. We prefer this one here. I'm just showing you one of the videos of my postdocs where we put cells in microgravity, and these cells are kept in free fall. It's like weightlessness, a constant fall. So our product is called Beyond Age. We claim that we can know your tomorrow, and this is the way the workflow goes. We have a vacuum tiner. That's one of the, I say, issues of bringing things to market. You need to make it user-friendly. This is not very user-friendly, and that's why we're partnering with clinics. We have a vacuum tiner. We need around 3 of those to make it happen. It's an expensive test. We give your cells a brief space vacation, and then with RNA sequencing, we report back in this app. So as a summary of this, simulated microgravity act as an aging stressor in brain, heart, and immune organoids at the level of the transcriptome, at the level of the functional responses. We can mimic diseases, especially Parkinson's disease, cardiomyopathy, and immunological aging. The tech is suitable for the screening of geroprotective compounds. The issue we have at the moment is not high throughput, because these machines have only 4 ports. So that's a limitation we have. We would have maybe a big warehouse full of these machines. We can scale, and we can do it high throughput. And then last, there's a robust application in longevity medicine as a way of getting into the market. And these are my emails if you want to reach out. Thank you so much for your attention. Questions? Yes, we have plenty. One there.
That's very interesting. Have you tested this in other species? So, for example, if you were to make organoids from more naturally aging-resistant species, naked mole rats, bats, whales, would they be able to resist it better?
We haven't tested it. What we're planning to do is to get samples from centenarians. And so, doing it in diseases, in other species is a great idea, but we're focusing on humans. And so the model that you're suggesting, the closest in humans, is supercentenarians. We haven't done it, but we're very close to getting samples. PBMCs, the 3 vacuum tiners full of blood from supercentenarians is not something super easy to get, but we're a nanometer away from getting there. Yeah.
David, great talk. You flashed one slide that particularly fascinates me, which is the trajectory matters slide. I wasn't familiar with the eye-pace biological clock, but it probably doesn't matter. So are you really saying that measures of biological age using epigenetic clocks are meaningless? It's the trajectory that matters.
I think the predictions are weaker if you use baseline only. If you use the trajectories, the predictions are much stronger because you're looking at slopes. The Dunedin pace of aging is the closest as we can get to that metric. But I don't think they're meaningless. I think it gives you, it's not I think, it does give you a prediction at a population level that is less accurate than a trajectory-based clock.
I mean, there are many things like genetic risk scores that work at a population level, but not on an individual level.
Correct.
And yet we assume that they work at an individual level.
Yes, fully agree. Question over there, in the back. Oh, here.
So there has been more discussion about essentially returning humans to space, settling the moon or Mars, and with that can come exposure to conditions in space, both in transit and on those new worlds, that could lead to accelerated aging through some of the phenomena that you described. do you have any ideas about how to reliably prevent that if we were to have more of a human population on the moon or Mars? How can we at least keep their aging to the rates of aging observed on Earth, if not slow it down further?
Yeah, well, so you need to get there first, right? And to do so, you definitely need a variety of different geoprotectors that you can take before flying, right? We're planning now to send astronauts in the next mission with Chris Mason at Cornell and give them different blends and see if we can prevent these effects from happening. Establishing colonies out there, basically you need gravitation. If you have gravitation, I think you're going to be in good shape. That's the highest risk, as I see it. If you go beyond low Earth orbit, there's also radiation. So it's a complicated question, but I hope I gave you kind of a decent answer there.
Yes.
Yeah.
Thank you. I was amazed again at your trajectory slide.
Yeah.
And in fact, looking at first glance, it seemed that you have several quite steep negative slope trajectories in there. Are those real? And what are those really? I mean, they really decline a lot. And there were at least 5 to 7 at a first glance.
Yes, it's
and
much
you didn't
more,
point them out, but what happened?
they're definitely real. All that data is real. It's longitudinal data from 1000 Immunes project. So you have those trajectories that happen over time. It's much more common than we think it would be. When you see the population, you have a huge increase, right, in the eye pace, which is expected. But I would say maybe 10% of people, which is massive, have either stable, no change, or decline, which is telling you that they're getting better for whatever reason. We haven't fully analyzed their nutritional status and the lifestyle, but it requires more. What's that? We don't know today what are the root causes of those outliers, but super interesting. Yeah. Thank you so much.